Okay, so thank you all for joining and thank you for the invitation. We are pleased to be here today. So in this talk, we're going to discuss our recent work on accelerated hashing-based data structures on GPUs and how we were able to leverage the power of these data structures to accelerate a selection of common bioinformatics applications. So in this first half of this talk, I am going to discuss how we were able to exploit the fast memory interface of modern GPUs by proposing a parallel hashing scheme, which is tailored to improve the memory access patterns on CUDA architectures. I would then like to give the floor to my PhD supervisor, Bertil Schmidt, who will talk about our advances in the field of genomics through the power of these GPU hash tables. So let's dive in. So first, what drove our research focus towards GPU hash tables? Well, TLDR on this is it's all about the memory bandwidth. For instance, on modern CPU workstations, we typically have memory modules with a couple of hundreds gigabytes per second maximum bandwidth. And if we have very deep pockets, we can afford maybe a few terabytes of these modules per node. Conversely, modern professional NVIDIA accelerator cards, which are equipped with on-chip stacked memory modules, they output up to 2 terabytes per second, but they come at a higher price point, so we can maybe only afford a few gigabytes per chip. So it's, in this example, 80 gigabytes for the flagship, which is an A100 accelerator. If we take a step back and look at hash table operations, they are in general memory bandwidth or latency bound, meaning that the throughput of the table is mainly determined by the performance of the underlying memory system, which makes GPUs a valid option for this purpose here. More specifically, due to the nature of the hash function or hash functions, we observe a random memory access pattern. Thus, we are bound by the number of random updates we can do per second, which is typically only a fraction of the theoretical maximum bandwidth you've seen before. However, we will show how our new hashing scheme is able to alleviate this constraint by improving the data locality during hash table probing. Okay, so why is hashing a good idea in general? Well, hash tables are associative data structures that can map an often sparsely populated domain of keys onto a value domain. In other words, they implement a key value store. This is achieved by mapping the sparse key domain onto a dense memory domain, so a location within an array, using a hash function over the keys, along with some collision resolution strategy, which is required since the mapping itself is non-injective. The main benefit here is that these steps do not depend on the overall number of elements residing in the table, which gives us an O of 1 time complexity for insertion and retrieval, respectively. Additionally, hash tables impose very little memory overhead, which makes them well suited for GPUs, since the amount of available video memory remains relatively scarce even nowadays. Some algorithmic alternatives to hash tables that achieve the same key value store semantic are, for instance, sorted arrays and a variety of tree-based approaches, both having logarithmic time complexities. Two things to note. So since in-place swap-based sorting algorithms are not very suitable for the GPU, the sorted array approach needs a total of 2n auxiliary memory. Tree-based approaches, on the other hand, additionally exhibit highly irregular data layouts that are hard to construct in parallel without any auxiliary memory or even dynamic memory allocations. One additional consideration, while sorted arrays and most trees support some sort of range query capability, hash tables, due to the random nature of the underlying hash functions, do not. So if range queries do not matter for your use case, then you should definitely take a look at hash tables. What are our contributions? Well, we proposed a new library for GPU hash tables, as well as other hashing-based data structures, which is designed around 4 conceptual pillars. First and foremost, our main focus lies on performance measured in throughput, meaning table operations per second. Warp Core outperforms other state-of-the-art CPU and GPU hash tables, which we will see in the evaluation section. Second, we have modularity. So our experience with GPU data structures over the recent years revealed that there is no one-size-fits-all implementation which works best for every use case. Thus, we split our implementation into several interchangeable modules, such as probing schemes, hash functions, or even memory layout. So, for instance, an array of structs versus a struct of array layout. This way, we can tailor the data structure towards any specific application in order to achieve best performance. Then third, we have a host and a device-sided API. So Warp Core provides a simple host-sided API for bulk operations, such as insertion or retrieval of millions of elements in parallel at high throughput. And then additionally, we provide a device-sided interface, so we can call table operations from within CUDA kernels, which can be used to fuse table operations with other tasks in a single kernel, which is a versatile concept to avoid expensive synchronization, so global synchronization between kernel calls, in cases where distinct CUDA threads or warps perform computations depending on values that come from the hash table. Note that the host-sided API or interface internally also uses these device-sided functions. And lastly, we have a very important thing is that all operations can be bound to and therefore synchronized with CUDA streams, which allows for asynchronous task overlapping and also enables us to utilize multiple GPUs. Let's take a look at the implementation. So first, let us discuss a general example on how hash tables and especially data parallel hashing works. Down here, we see the memory footprint of a hash map with, in this case, 16 slots for keys and the associated values in either array of structs or structs of array layout. Excuse me. It's basically just an array of length 16 here. The hatched position denote already occupied slots in the map. If a single thread, here denoted as A, attempts to insert a new key value pair, Ka and Va, into the table, it first applies a hash function, h, to the key, which gives us a mapping from the key domain into the index domain of the array. In other words, we will get an initial slot index from this hash function. If this slot is unoccupied, we can simply write a pair to set position and we're done. However, if this initial slot is already occupied by another pair, meaning we observe a so-called hash collision, we have to apply a suitable resolution strategy for this, which I will discuss on the next slide. Also, if we think of a multi-threaded setup, multiple threads may write to the same slot concurrently, resulting in a race condition. In order to circumvent such illegal states, we make use of atomic instructions, namely compare and swap operations here. So if 2 or more threads attempt to insert a pair into the same slot, the atomicity of the cas operation guarantees that only one of these threads succeeds, while the other threads need to follow the aforementioned collision resolution strategy. Note that retrieval of an element works in the same fashion. So we simply apply the hash function to a desired key, check if the associated slot is occupied by a said key, and subsequently return the value. Collision resolution strategies. Well, there are 2 main approaches to resolve said hash collisions. The first approach is called separate chaining on the left, which bypasses this problem entirely. by collecting all colliding elements, so keys, inside a so-called bucket for each hash table slot. These buckets can either be pre-allocated arrays or even dynamically allocated linked lists. In the first case, implies that we have to over-provision each bucket with enough slots to hold all colliding pairs, which is frankly not known in advance. So when we build the hash table, so we have to do a lot of over-provisioning here, which is not very memory efficient. In the second case, well, linked lists need dynamic bucket allocation or list allocation, which also degrade performance, especially on a GPU. The second main approach for correlation resolution strategies is, which we focus on in this talk, because it's much more suited for CUDA architectures, is called open addressing. Here, the table itself is a linear memory segment, so it's just an array, which is not subdivided into distinct buckets. If a collision occurs, we simply move to a different slot determined by a sequence of alternative indices for that specific key. These sequences are called probing schemes. There are three main schemes falling into this category, which are called linear probing, quadratic probing, and double hashing. So the simplest open addressing probing scheme is linear probing, where we simply move to the next neighboring position until we find a suitable slot. This is obviously very cache efficient because we simply probed through a whole cache line, but linear probing suffers from a thing called primary clustering, which are basically ever expanding dense regions inside the table. And we have to probe through these dense regions, which slows down probing significantly. Quadratic probing tries to lessen these clustering effects by leaving dense regions faster, so quadratically. However, there arises a second problem. So if two distinct keys collide at the first probing position, then with quadratic probing, they also follow the same probing steps. This effect is then called secondary clustering. Then as a third option, we have double hashing, also often denoted as chaotic probing. As the name indicates, it's the most cache inefficient scheme because we're simply seemingly randomly jumping inside the hash table, but it shows none of the before mentioned clustering effects. So we have a very uniform table occupancy. Note that there are also schemes that rely on swap operations, for example, cuckoo hashing and Robin Hood hashing, which have their theoretical benefits in terms of average probing length and so on, but they impose many undesirable constraints on the table, because if we need to swap elements, then yeah, it's hard to synchronize this with other parallel table operations. Yeah, so we won't focus on this. What we do is, so our approach basically combines the cache efficiency of linear probing with the uniform table occupancy of double hashing. So we don't encounter these clustering effects, but have the cache efficiency. So how can we do this? Well, as I said, hash table operations experience random memory access patterns. And this effect is even more pronounced in CUDA accelerators. So we propose a cooperative probing scheme specifically tailored towards the CUDA architecture, which aims to reduce the number of cache misses per probing step by looking basically at multiple adjacent slots in the same cache line in parallel before moving to another outer probing window, which is determined by a double hashing scheme. So we have inner linear probing and outer double hashing. But instead of using a single thread per input element, we utilize a so-called CUDA cooperative group, which is a group of coalesced threads in a warp that can share the same cache line and are able to communicate via fast registers using warp intrinsic operations such as shuffles or vote operations. So here in this example, we use a group of 4 coalesced threads to insert a new key value pair into the table. First, we determine an initial probing window by applying a hash function to the key, which we see in step 1 here. Subsequently, each thread in this group copies the contents of its respective slot from this window into a local register, resulting in an efficient coalesced load operation within the same cache line. The group cooperatively determines which slots within the window are unoccupied. This is realized using the before-mentioned vote operations. Here in this example, in this window, all slots within the window are already occupied. So we move to the next probing window according to the outer double hashing probing scheme. Here, the group determines 2 candidate slots following an inner linear probing scheme. So from left to right, we try to swap the new key into the first candidate slot. If successful, this means that the new key is now resident within the table, within the slot. In this case, we can write the associated value and just successfully exit the operation. However, if this CAS operation fails, then the state of the slot has been altered since step 5. In this case, we move to the next candidate slot or even to the next candidate window. In short, we can say that the outer intergroup chaotic or double hashing probing scheme ensures a uniform table occupancy, so no clustering effects again, while the inner intragroup parallel linear probing is cache efficient. Oops, sorry. All this works fine if any distinct key occurs at most once in the input data. So we have a so-called single value scenario, so where all keys residing in the table form a mathematical set. However, if we want to associate multiple values with the same key, we need to consider a few additional things. So this is called a multi-value scenario, where a key can occur multiple times, a distinct key can occur multiple times inside the input data set. So for once, the probing scheme has to be cycle-free in order to guarantee when retrieving every stored value that we do this exactly once, because if we have these cycles, we might find the same value over and over again. Since we are using double hashing as our outer probing scheme, we can guarantee this by simply choosing the table capacity to be prime. This is just a theoretical nice thing that double hashing gives us here. Also, we can then utilize the cooperative group to retrieve multiple values in the same window in parallel. So again, we can fully utilize these cache lines during probing, which we'll see on the next slide. Hahowever, one problem remains when using this open addressing variant for multi-value scenarios. The hash map is forced to store identical keys multiple times, which is very memory inefficient, especially for highly skewed data distributions, where maybe a distinct key may occur hundreds or thousands of times, and we have to store this key each time in the table. So in order to improve the space efficiency for such skewed data distribution, we also propose a new bucket list hash table format. So here, keys are stored only once inside a single value open addressing hash table, as we've seen before, while the associated values for each key are resident within a linked list of arrays called buckets, which can have varying sizes in order to adapt to the input data distribution. For each occupied slot, the single value hash table holds a handle pointing to the tail of its associated bucket or bucket list that can be updated atomically. So this is basically the value associated to the key in this single value hash table. This is the pointer, basically. During insertion, new values are simply appended to this tail bucket. The handle always points to the tail bucket, so during insertion, we don't have to chase this bucket list back to the head or from head to tail, but we simply grab the last bucket, the tail bucket, and insert the new value there. If a bucket gets full during an insert operation, we request a new one from a pre-allocated memory pool with a size determined by a user set bucket growth factor that can be tuned towards the input data distribution by reducing the space occupied for each bucket link. This approach basically can be seen as a hybrid between open addressing and separate chaining. Let's take a look at an example to make this a bit more clear. So this rather fancy illustration shows the parallel retrieval of all values associated to keys K1, K2, and K3 from a bucket list hash table. And this is performed by 3 distinct cooperative groups here in parallel. The upper memory segment represents the key store, so a single value hash table, while the lower array denotes the value store, so the pre-allocated memory pool already populated with buckets that are potentially stored out of order depending on when they were allocated. Following the cooperative probing scheme, we initially look up each key from the store. Subsequently, we take the handle that is associated to this key, which points to the tail of each buckets list. Then the cooperative group iterates over all buckets within the list in parallel. So again, this is very cache efficient. We can basically do coalesce loads from these buckets here. So parallel lines in the lower part of this depiction indicate an efficient coalesced memory pattern. Okay, then let's dive into some evaluation here. So we benchmarked our open addressing single value hash table against the publicly available state-of-the-art GPU implementation SlabHash, which uses a variant of separate chaining, and also cuDF, which is an open addressing linear probing approach, which is part of NVIDIA's RAPIDS framework and is purpose-built for performing hash joins between cuDF data frames, basically. Our benchmark scenario here consists of an initial bulk build operation, which inserts a set of 2 to the power of 28 unique 4-byte keys along with 4-byte arbitrary values into each hash table. And this is done on a single V100 accelerator card. So this is the insertion step is shown on the left, and on the right, we query the same set of keys against the table and retrieve their corresponding values and report the resulting throughput for a specific target occupancy, or also called storage density. The 2 black lines in each plot correspond to warp core, where the distinction between these 2 lines is the size of the cooperative group used during probing, 4 or 8 here. Here in this setup, we see only slight differences between these 2, where a CG size of 4 seems to be maybe a bit faster than 8. However, we will later see in which cases the size of the cooperative group has a higher impact on performance. Okay, so for insertion, we see that our best competitor, SlabHash, is maybe on par with warp core for load factors up to 70%. However, if the target density increases, then WarpCore's cooperative probing scheme is superior. For retrieval, WarpCore consistently outperforms the other implementations while also maintaining robust throughput even at higher load factors. And also, if we compare WarpCore against cuDF, we see that the linear probing scheme from cuDF does not really cope well with the higher load factors due to the aforementioned clustering effects. Sorry. So now that we've seen how these hash tables perform under a unique input distribution, so we have seen a single value scenario, let us now switch to a multi-value scenario. In this case, we have an average key multiplicity of 8, meaning that each distinct key in the input data occurs roughly 8 times. This next multi-value experiment is designed to elucidate the effects of average value. I'm sorry. That's a different slide. So here we compare our open addressing implementations to the new, more memory efficient bucket list approach, which is shown in blue. And Also the only competitor which we know of that supports multi-value scenarios out of the box, namely QDF. So our initial observation is that our open addressing implementation follows a similar trend compared to QDF due to the aforementioned clustering effects of QDF's linear probing scheme. The performance really degrades drastically when the number of values is increased. Here, these clustering effects are even more pronounced since identical copies of the same key will get hashed to the same memory regions in the map, resulting in even larger dense clusters. In contrast, here our new bucket list approach shows almost identical throughput for every run. Our explanation here is that since the number of steps required for inserting a new pair into the bucket list is independent from the number of values already associated to the key, while the number of required probing steps in the open addressing tables increase with the value multiplicity. One additional thing we observe here is that now that the key multiplicity is higher than in the previous setup, we see that WarpCore's open addressing approach consistently performs better with the larger of the two group sizes. This effect is even more pronounced if we increase the key multiplicity even further, for example, by using an exponential input distribution. So the limited amount of main memory available on a single GPU can be insufficient for many data-intensive applications. Thus, WarpCore allows for distributing hash tables over multiple GPUs connected via a dense NVLink topology. As an example, on the right, you can see the NVLink topology of a DGX1 Volta server, which features 8 V100 GPUs shown in green here. Let's say you have a set of key or key value pairs that are randomly distributed over all these GPUs, so basically scattered over into the global memory of the GPUs. On the left, you see such a layout for, in this example, 4 GPUs, and here's 7 keys per device, so 28 in total. Oops. The first step to take is to decide which of the keys belong to which GPU. This can be achieved by applying a small hash function to each key and take the modulo number of GPUs. We arrange the data then in memory so that keys that belong to the same device reside in a continuous memory region. And each GPU does this so-called multi-split step in parallel, and you can see the outcome of this operation by the color coding here in the second depiction. Subsequently, each device then sends these chunks to each target GPU asynchronously over the NVLink topology. This is called an all-to-all communication scheme here. The result shows that we now have successfully partitioned our input data, so previously randomly scattered input data, now partitioned over all devices. So from here, each GPU simply maintains its own WarpCore hash table and inserts or queries the elements against the table. This is also published work by us, which is called the Gossip Communication Library, which basically performs this all-to-all transposition over NVLink. And we achieve up to 1.8 terabytes per second on a DGX2 and a half a terabyte on a DGX1 Volta. So as a benchmark here, we simply use 2 to the power of 27 unique 8-byte keys and values as input per GPU. So in total, we have 16 gigabytes of data per GPU. This plot here shows a weak scalability analysis with runtime breakdowns for data partitioning. So that's the multi-split phase, the communication, that's the all-to-all transposition over NVLink, and the hash table operation itself. In this case, it's an insert operation. And also the black line shows the achieved weak scaling efficiency. Note that it would be possible to further apply common optimizations like batching or overlapping CUDA streams to hide the runtime of data transfers behind kernel execution. for instance, for multi-split and insertion, but we decided to report the full write runtime for each primitive to show the relative cost here. And what we see is we achieve 100 gigabytes per second throughput using 8 Tesla V100 GPUs, which corresponds to a scaling efficiency of 53%. Okay, so that's it from my part. I'd now like to give the floor to Bertil Schmidt. So I stop my presentation real quick.
Okay, so in the second part of this presentation, we are looking at applications of our hashing library to bioinformatics, in particular to sequencing, DNA sequencing data sets. And this is motivated by the rapid development of sequencing technologies, also called next generation sequencing technologies, which use samples of DNA or RNA and then output the genomic string of the input cells. So these technologies, important to know now is that these technologies do not output the full genome strings, but just substrings of this genomic string. And these substrings are typically, depending on the technology we are using, but in case of Illumina technologies, around 100 to 150 letters or base pairs in length. And to cover this whole DNA that was sequenced with these short fragments, you have a huge redundancy of these strings. So you normally output hundreds of millions or billions of those short strings called reads in a single experiment. As you see in these diagrams that I have on the bottom left and bottom right, first on the bottom left, you see that the sequencing costs have been decreasing dramatically over the last few years, over the last decades actually, and we are now, depending on the sequencer that you are using, even down to around $100 per human genome sequencing, which are the costs. This is based on Illumina systems. There are also other systems that you can use with a bit higher costs. And also the amount of produced data is, of course, growing rapidly in this area, as you can see on the lower right-hand diagram here. One very important and very interesting application area of this technology is metagenomics. In metagenomics, you take a sample that you put into the sequencer from an environment. That can be different environments. Examples include soil, gums, air, and also food, what we will be discussing later. And you now normally do not know from which genomes are actually in your sample, and you actually want to find out what genomes and to what quantity these occur in your sample. So the reads that you get out of your sequencer do not stem only from a single genome like a human genome, but they come from a mix of different genomes which you do not really know beforehand. And this leads to the so-called taxonomic read assignment problem. So for every short substring, so every read that comes out of that sequencer, you want to find out its true origin. So from which genome does it come from? And once you found that out, you can actually do that for every read in your sequencing data set. Then you can kind of deduce what components are, what things are actually in the sample that you have sequenced. And so this task includes then two data sets, right? So a data set of those reads that come from your sequencer. And in order to try to find out the taxonomic origins of these reads, you compare them to a huge database of reference genomes. And so this requires two big data sets and a comparison of these reads to these reference genomes. And this can be done very well with those hash tables that we have seen on the previous slides and with the WarpCount library on GPUs. We actually started with that several years ago as a collaboration with the biology department at our university, collaboration with Professor Tom Hanken's group, where we sequenced food, in particular sausages, as you can see here, kind of sequenced sausages. And then we get out reads from these food samples, and then we compared them to various genomes. At that time, in our first pipeline, we just used standard alignment tools, like this Q-short tool here, and also BLAST, and also Megan. So these are tools that do kind of full alignments and are very slow. So this pipeline kind of could find out the ingredients of these sausages that we sequenced, but this was running extremely slowly. So we were looking at methods that can do that in a much quicker way, orders of magnitude faster. And the first idea is here not to use actually expensive alignments, but to use just comparison of k-mers. So k-mers are short substrings of length k of a read or of a reference genome. Here you can see an example. So you have, let's say, a read here, CGTAA, and so on. And in this diagram, I extracted all k-mers with k equals 5. So all substrings of length 5. And now you use all substrings of length 5 to query a database. And this database is a key value database, as can be implemented by hash tables, with a k-mer as a key and the value taxonomic IDs where this k-mer occurs. So taxonomic IDs meaning genomes that have that k-mer in their genomic string. So in this approach, that was the first approach that implemented that several years ago. It's called Kraken. That takes every k-mer of a read, queries this database, gets out taxonomic IDs, builds a taxonomic tree, and then looks at high values in this taxonomic tree, like this orange 5 here, to classify that read as a certain genome. So this approach proved to be much faster than these alignment-based approaches. It's relatively simple. The disadvantage of this approach is that this k-mer index for big databases is far too big. So in Kraken, for example, several terabytes in size if you do it in this way. So normally it doesn't fit on the main memory of your machine. So that's why we developed a subsampling scheme that reduces the amount of k-mers that is stored in this table, thereby producing a much smaller hash table that can fit on the main memory, let's say of a single GPU, or if it really doesn't fit anymore, then you use this multi-GPU system to have a distributed hash table. Also can be done with WarpCount. And the subsampling scheme is called a locality sensitive hashing scheme. In this case, we use MinHashing. MinHashing provides an approximation of the Jaccard index of two sets. And it applies a hash function that is applied to all k-mers in the set, and then produces a sketch. The sketch consists then of the S smallest hash values. So of the k-mers with the S smallest value of your given sequence. And this is then just a subsample of your original string, thereby reducing the amount of k-mers that is stored. Here's an example. I have two strings, and these strings are different at one position. This red T in the first string and the red A in the lower string. The rest are, other letters are the same. And here k equals 3, so we extract all 3-mers, apply this hash function and create an integer value. And you can see from the integer values, since most of the k-mers are identical, these 3-mers are also identical, except for these 3 k-mers here that overlap with a character that is a different character. These then produce different integers. And now you can, one way to compute the similarity between those two strings is to compute the Jaccard index of these two sets, AH and BH. When we now use MinHashing, we reduce the k-mers and then compute the Jaccard index of the reduced set. Okay, in this case, for example, to 4. So we just use the 4 smallest values of the set to compute the similarity. This case, you get then 0.6 as the similarity, which is quite close to the full similarity of this, which is, I think, 0.57. So this can be also shown to be a good approximation of sets, MinHashing. And this scheme is used, MinHashing, in connection with a warp core to produce our tool, which is called MetaCache, which is a tool to classify metagenomic reads and also to output quantification. So the percentage of different organisms in your actual sample. Metacache-GPU consists of 2 phases, a build phase and a query phase. In the build phase, you take every reference genome that you consider and put it and build a hash table using WarpCount. Okay, so you take the reference genomes, partition every reference genome into windows, extract all the k-mers, do the MinHashing, and then you put every k-mer, MinHashed k-mer, into that hash table. So you get a multi-value hash table where you have a key, which is your k-mer, like let's say this blue k-mer, and then all the genome and the genomic locations where this k-mer occurs. Now in the second phase, you have the query phase. You take every read, do the MinHashing, and then use the k-mers to query that hash map, and you retrieve the genomic IDs where this k-mer occurs. You do that for all the k-mers in your sketch, and then you get counters, and the genomes with the highest counters are then chosen to classify your read. Let's say this one has, the genome number 1 has 12 hits, so this read might be classified as genome number 1. So this Metacache-GPU version was evaluated first in terms of accuracy. So when we used our sausages that we sequenced, could we really find out the ingredients of the sausages? Here is one example. We had many sausages. All these sausages were called calibrator sausages. So in these calibrator sausages, we collaborate with a butcher, and this butcher prepares the sausages according to a fixed recipe with exact amounts of meat and other ingredients. And then we sequence that sausage, get out these reads, and then we run our Metacache-GPU tool, and then we find out what ingredients come out of Metacache-GPU and compare them to the ground truth ingredients that the butcher has used. Here's an example of a sausage, which is a horse sausage. Yeah, I know that, I mean, horse sausages are not that common, but you can still buy them at different markets, also in our region. And so this sausage has had 55% horse, and Metacache-GPU found out 54.8% horse, which is quite close. Pig is a bit underestimated, 31% pig. And the ground truth is 35% pig, which mostly pig is often underestimated because it contains lots of fat. So if it contains a lot of fat, kind of less reads will be sequenced. And also 1% cattle, which we also found out quite well, and some sheep. Okay, and again, there are many other sausages that we also tried and could classify quite well. In terms of runtime, there are different components, of course, and here I just want to draw attention to one of the components of the runtime, which is the most time-consuming part, which is database construction. In this database construction, we used 151 gigabytes of genomic data and extracted the k-mers and put them into this massive database. And we compared the runtime of the multi-threaded CPU multi-threaded Kraken 2 tool, then our MetaCache tool executed on CPUs, again, multi-threading, and our MetaCache-GPU tools with 8 GPUs on a DGX1 V100 server. And you can see here massive speed up. So Kraken 2 requires 4 hours, MetaCache CPU 3 hours. And from hours to less than minutes. So with the MetaCache, you can actually do that at 43 seconds, which is huge speed up here. The second application is called k-mer counting that I want to discuss. In k-mer counting, you again have the set of reads. So millions, billions of those reads, and you extract again all substrings of length k, and you just count the occurrences of each substring of length k. So you have a quite simple example with 3 reads, k equals 4. This would then be the output kind of the k-mer counter. ACGT occur 3 times, CGTT 3 times, GTT twice, GTTT 1. So this is a k-mer counting problem. Very important procedure in tons of bioinformatics applications. Here I just mentioned a few. But this is used very frequently, and there are tons of tools that do that. Mostly on the CPUs, there are now a few GPU tools, and of course, with WarpCore, you can easily implement that. In the WarpCore, then you have a single value hash table, and in the single value hash table, when you occur the same k-mer, you just increment the value every time. So a counting hash table. So can be implemented relatively quickly with such a library. Here a bit what you can expect as an output from this k-mer counter, a k-mer abundance histogram. Here you have k-mer frequency and number of distinct k-mers on the y-axis, k-mer frequency on the x-axis. And you can see that many k-mers occur 100 times, that many k-mers occur 500 times, and so on. From this histograms, you can very quickly notice, for example, cross-contamination in genomic sequencing projects, where you have, for example, 2 Gaussians. When you have 2 Gaussians, like in the upper right diagram, you can have, it's very likely that your sample is contaminated in the case of haploid organisms. So our WarpCount, WarpCount is a k-mer counter implemented with a WarpCore library. You start on the GPU, passing your input file, and then sending it to the GPU. On the GPUs, you extract all k-mers. And in the case of a multi-GPU implementation, as Daniel told you earlier, you have a multi-split and an all-to-all, and then you can use counting hash tables. In addition, we use Bloom filters here to remove k-mers that occur only once. And then these results are retrieved and written to a file on the CPU. This is a performance evaluation of WarpCount for 3 data sets in comparison to the state-of-the-art CPU, multi-threaded CPU versions, Jellyfish 2, DSK2, and KMC3. And we can, again, achieve very good speed up. This is based on a single V100. And we get a speed up of up to 13 compared to KMC3, which is kind of the state-of-the-art CPU tool here. Okay, I think that's then the end of our talk. Just as a summary, we have presented the WarpCore library for GPU hash table data structure, which is a framework for high throughput hashing data structures that can be tailored to fit many use cases. We have efficient single and multi-value hash tables. What was not shown here, hash sets, counting hash tables, and Bloom filters also included. The multi-value hash table approach is new, can scale to multiple GPUs, and can be applied for many applications, especially in bioinformatics. Okay, and this is available on GitHub. So thank you very much, and we are happy to answer some questions, I guess.
Thanks a lot, Bertil and Daniel, for this interesting presentation. Are there any questions? Please raise your hand if you have any questions or comments. Tobias.
Yes, thanks for the really interesting talk. I wanted to ask how well these kind of techniques would be transferable to the setting where you have multiple hash tables for different parts of your data you need to update. Some of them maybe even residing in shared memory, like in local fast memory. So how well do these techniques rely on having like a global memory architecture? And can you also scale down your warp processing, for example, to subwarps or smaller units?
Yes, thank you for the question. So I'd like to split the answer up here. Okay, so yes, we can use subwarp groups. That's the default, actually. So by default, WarpCore uses a group size of 4. So you have a warp of size 32 and you divide it into groups of 4 threads. But you can set this as a parameter. That's not a problem. Second, if you think about having multiple hash tables, that's also not a problem. So you can think of this as a standalone object with data in global memory. So as long as you have enough global memory left, you can simply start multiple hash tables or construct multiple hash tables, and you can also bind them to different streams. So all the operations, you can bind them to different streams. So you can use them asynchronously. The last part of your question was, is shared memory an option? So for WarpCore itself, it's not. It's just a global memory thing, but we are currently working with NVIDIA, who, so I had an internship at NVIDIA, and what we did is we basically incorporated parts of WarpCore into a new hashing library, which then also supports shared memory hash tables. You can either build them in shared memory and use them from there, or you can have a global memory hash table and then basically get views into shared memory, copy parts of the hash table into shared memory and use it from there. So that's the thing that's coming up in the near future.
I have a question. As far as I understood, in your performance benchmarks, you used keys and values which were 4-byte integers, right? For the benchmarks. Does it make any difference at all if, for example, the values are more complicated objects? Not just integers, but like structures or any type of data?
So if we only...
a good question.
Definitely.
Yeah. Okay, Daniel, you can, let me talk first. You need atomic operations. And at least for the key, for the values, probably not such a big problem. But you only have 32-bit and 64-bit. So actually we use 4-byte and 8-byte in our performance evaluation. For the key is quite difficult without atomic operations. And so NVIDIA GPUs are currently only 32-bit and 64-bit atomic operations. Yeah, but Daniel, I think for the value, you can be a bit more relaxed, right?
Right. So for the values, you can have larger, more complex values as long as they are trivially copyable. So I can simply copy them into the hash table slot, right? But a thing that needs, or you have to think about, is the memory alignment. So yes, we can have 32 and 64-bit native atomic support for keys on CUDA, but let's say you have a array of structs layout where you basically have key value, and so on, then the CUDA architecture requires you that the alignment of the keys you want to write atomically is properly aligned. So 64-bit for 8-byte types. One thing we can circumvent this whole thing is by using NVIDIA's libq++ library, which is basically a new wrapper around the STL C++ library for CUDA GPUs, which features a dedicated CUDA atomic type, similar to the STID atomic type, which takes care of all this stuff. So I can basically insert a pair, a key value pair, make this CUDA atomic, and it takes care of the alignment for you.
Can you just pad accordingly? I mean, if you align the key to 64 bits, and then whatever comes after that, and then you add an appropriate amount of padding so that the next address for the next key is aligned. Wouldn't that be the simplest solution?
Exactly. So that you can do this by hand, or the CUDA atomic library will take care of that for you. Okay.
Okay. Another question: the bandwidth numbers that you reported, the 100 gigabytes per second on 8 V100s. How's this bandwidth calculated? What's the actual bandwidth you're measuring there? It's not the memory bandwidth of the whole system. It's what is the formula for determining this bandwidth?
It's basically you have the size of your input data set. So basically add up all the numbers of key value pairs, the sizes on all GPUs, and then basically measure the time it takes for the data in GPU residing in GPU memory to build the hash table from them, and then that's basically the throughput. So input size divided by time.
Okay, so the actual bandwidth going over the memory bus is a multiple of that, because you're not just touching everything once, okay? At least you read and write, something like that. So what is the actual bandwidth going over the memory bus? That would be very interesting, because you said in the beginning that everything is either memory bound or memory latency bound. And it would be interesting to see how far away you are from the memory bandwidth barrier, or how large the impact of latency actually is.
So the speed of light, actually.
Yeah.
Right. So we have a few more numbers on this in our gossip paper. So on the NVLink all-to-all transposition, where we do an analysis on that. I cannot remember the actual speed of light there right now. I need to look it up. Bertil, do you know it maybe?
Okay, I think you're talking about something different. In the gossip paper, it's more about the GPU to GPU communication. But what Georg was asking on the single GPU implementation, the bandwidth to main memory, what is it? What is the bandwidth that we are getting out of that transfer? Not GPU to GPU.
All right. Thanks for the clarification. To give an intuition on that, so when working for NVIDIA, we found out that Warp Core achieves around in the mid-90s% of speed of light in terms of random update performance, memory updates on an A100 GPU. So it's just one number I have there for that. So it's pretty close to optimal. Sure, there are some things that can be optimized, but it's like 92 or 93% speed of light there.
Thank you again for the presentation. Bye-bye, everyone. See you next time.